Okay, let's go ahead and finish this week off. So exams are down front. They'll be in the lab for anyone who doesn't pick them up today. Exam went pretty well. Average was just a couple of points lower than on the first exam. Usually drops a little bit more than this. People did really well. Check to make certain that I added back the proper points. So I threw out 5 questions because more than half of you got them wrong. So those points were initially taken off and then added back. So if you see a plus, 6 or 8 at the end, that should be the number of questions that you add back. So make sure that I managed to get all of them back. And always check my arithmetic because, yeah, after doing about 30 of these in a row, I get pretty stupid. So anyway, I'm going to try and keep the lights up this time because a couple of people told me they keep falling asleep when I turn the lights down, and it is Friday, so hopefully people can still see the screen okay with the lights up. I prefer the lights down, but, I don't want you to fall asleep. Well, you're going to fall asleep anyway. But. So we left off on Wednesday talking about the Agrobacterium tumefaciens system. So this bacterium that has the capability of causing a neoplastic or cancerous disease in plants. Non-lethal cancerous disease, but cancerous nonetheless. This is actually the best slide to illustrate this whole process because this slide has everything, right? So anyone who's interested in microbiology or diseases, this has it all. We have 2 component systems shown here on the top. So remember that I said that the bacterium, Agrobacterium tumefaciens, as long as it has this TI plasmid, can sense wounded plants in the environment. So it's sensing phenolic compounds that are being given off as plant signals and as a result of wounds. And so the organisms will now detect using a sensory histidine kinase those plant phenolic molecules. They will phosphorylate a histidine residue on the cytoplasmic side, just like you studied for the last exam, transfer that phosphate group to a response regulator, which will now affect the gene transcription from the virulence region of the tumor-inducing plasmid. So this is a beautifully evolved system because it only bothers to express the genes necessary to cause the tumor in plants when the plant is there. So some of these virulence-related genes, all with the precursor, in terms of the nomenclature, always having the 3-letter prefix vir. So vir-D is one protein, vir-D2 is another protein. So some of these, and I don't care that you know what each individual protein does because honestly, I don't know what each individual protein does. The nice thing about this slide is that it does show you that there are a large variety, quite an array of virulence proteins that have varying roles within this process. Some of them are going to initiate a form of rolling circle replication. So very similar to what we talked about with the F plasmid. But now what's going to happen is that rolling circle replication is going to become arrested at a certain point. So the bacterium is going to initiate a rolling circle replication at a particular site, let's say on the right-hand side of this T, or transferred DNA, and it's also going to nick that same strand at the left-hand end. So that now when rolling circle replication takes place, what's peeled off from this is just a single strand subsection of the plasmid. So this differs from the F plasmid rolling circle replication, in which the formation of new DNA would simply go all the way around this circle, peeling one strand off while copying the remaining strand. In this particular case, the DNA polymerase is going to copy this inside strand, replacing the strand that it's peeling off. And so what's peeled off from that is a single strand piece of DNA, yet the plasmid is maintained. It's not lost during this process. This single strand section of the plasmid is now going to be bound. So it requires a whole array of chaperone proteins in order to make this next trip. So it's going, into a completely different domain of life. And so think of, if you're going someplace really strange and really different, there are a lot of things that you want to pack to make your trip better, safer. And this is what the tDNA is doing. It will become associated with various proteins like VIR-D2, VIR-E2, as well as VIR-F. These chaperone proteins will literally coat that tDNA so that you see as it's passing through this conjugal or sex pilus into the plant cell, the DNA, that single-stranded piece of DNA, is protected by a lot of proteins that were all encoded on this tumor-inducing plasmid and are now chaperoning this DNA through this type 4 secretion channel. So this type 4 secretion channel is very much akin to the sex pilus, the conjugation pilus that we talked about in the F plasmid. All of the proteins necessary to create this pore, as well as the machinery that's going to actively drive, by ATP hydrolysis, actively drive this nucleoprotein complex across here. All of those proteins are encoded in this TI plasmid, this tumor-inducing plasmid. That's why I say it really makes you think about the virulence of this bacterium, because the bacterium itself is virtually harmless. It's playing almost no role. It's a zombie. It's under the control of this TI plasmid. So this piece of DNA, now along with a whole host of accompanying chaperone proteins, some attached to the DNA, some free, are going to shepherd this tDNA ribonucleoprotein complex into the cytoplasm of the plant cell and actually get it past the nuclear pore complex of that plant cell. That's what NPC stands for. So this complex of DNA and protein makes it into the nucleic acid, and then some of those proteins that it's carried along with it are actually going to facilitate the insertion into the plant genome. It's a random insertion into that plant genome. Now, this is a really cool system, and even if the story ended here, and it doesn't, it would be a really cool story. But this process has been co-opted by people in biotechnology companies. This is the underpinning of every transgenic plant ever made, so far as I know. What people have done when they want to introduce a gene, say for an insecticide, into a plant like corn or something like that, they can do an infection, but what they do is that they genetically engineer this tDNA to carry the gene that they want it to. And so now you simply have to infect the plant that you want to introduce this gene into and just let this tumor-inducing plasmid do its thing. What's been taken out, obviously, are genes such as the cytokinin gene and the auxin gene. So in the normal plasmid, in the normal TI plasmid, as it has evolved, it carries with it copies or versions of the plant hormones, cytokinin and auxin. But these growth hormones are now going to be produced in the plant cell under the control of this tDNA that's been inserted into it. And now it's going to be expressed not under the normal control of the plant, when auxin and cytokinin are normally produced to cause cell division, but now it's just going to be produced full-time at the highest rate possible. So the cells are going to divide and divide and divide, and each time those cells divide, they're copying this tDNA. But then look at the other things, and I've already mentioned this a little bit, but I want to repeat it just to make sure people really get why this is such a cool system. Within that tDNA that's now being amplified every time that cancerous cell divides, there is the ability of that cell to now produce opines, these modified amino acids that the plant cannot use. So the plant makes them, but it can't do anything with them, so they build up to high levels. They're actually transported out of the plant cell, where the bacteria back home, carrying the TI plasmid, can use the opines as a carbon and nitrogen source. But the only reason that bacterium can use these very unusual amino acids is because other genes that are on the TI plasmid encode the entire biocatalytic pathway to break down opines. Normally, Agrobacterium tumefaciens without this plasmid If you gave it a ton of opines, it would just be completely confused. It wouldn't know what in the hell to do with them. But with this plasmid, it can break them down and use them just fine. So this is a very complete genetic parasite. So these plant growth factors are what's going to cause basically that tumor. It's not a tumor that we think of as like a virulent tumor, a malignant tumor. It doesn't metastasize. It just gets larger and larger. So this is a great system. It's just in terms of scientific knowledge, but it's also important that you understand this, because as you think about genetically modified organisms and plants and things like that, now you understand how technologically we can move genes into plants. Okay, done with Agrobacterium tumefaciens, but kind of not really. So what I'm trying to do here is to set up a comparison and contrast. So now I want to talk about the life history of a closely related bacterium, also a soil family of bacteria, that has some, what I hope you will see are some very obvious similarities to Agrobacterium tumefaciens, but instead of being a parasite, instead of being a pathogen, as Agrobacterium tumefaciens with that TI plasmid is, this family of bacteria, known as also within the Rhizobacteriaceae family, are actually mutualists. This is a group of bacteria that will interact with plants to help plants get a nitrogen source, to fix nitrogen. So nitrogen fixation, and by definition, nitrogen fixation means to take atmospheric dinitrogen, which is absolutely of no use to you or I. It's biologically inert as far as you and I are concerned. We can't do anything with atmospheric nitrogen. But bacteria, certain bacteria, and only bacteria, have the biological capability to turn atmospheric dinitrogen into ammonia. That is nitrogen fixation. Because various organisms, including ourselves, can use ammonia as a nitrogen source to synthesize amino acids, to synthesize pyrimidine, purine bases, and things like that. So nitrogen fixation is incredibly important, and the ability to do this lies entirely within the bacterial domain. Now, we're going to talk about one specific but very important subset of nitrogen fixation, and that's symbiotic nitrogen fixation. So this is fixation of nitrogen by bacteria in association with a plant. This is not to say, now don't walk out of this class thinking that all nitrogen is fixed by bacteria in association, in mutualistic association. There are free-living bacteria that are, excuse me, in the environment, and especially important in marine systems, at being able to fix nitrogen, to make nitrogen available, especially in marine systems. But in terrestrial systems, especially in the soil, probably the greatest source of biologically usable forms of nitrogen come from this type of a symbiotic nitrogen fixation that we're going to talk about. This kind of nitrogen fixation occurs predominantly in plants that are known as legumes. So you all know what legumes are at some level. These are peas, beans, but also there are a lot of plants that you might not recognize as legumes. By definition, legumes, here's a botany lesson for you, legumes are plants that bear their seeds in pods. So when you see a plant with its seeds in pods, that's a legume. And oftentimes these legumes are associated with, mutualistically with bacteria. That's why things like legumes and also lichens are very good pioneer species. So think about, and so as we talk about global warming, one of the big problems we know with global warming is that glaciers are retreating. So as glaciers retreat, the soil, that gravelly soil that's left behind there, is very nutrient poor. It's been buried. It hasn't had any organic material added to it for ages. Some of the first trees that will grow in that are alders. So alders are a type of tree that actually is associated symbiotically with nitrogen-fixing bacteria. So even though that soil has very poor nitrogen, or practically no nitrogen, there will be a nitrogen-fixing bacteria associated with the roots of those alder plants to help them pioneer the environment. And then as they break down, as they die, other organisms can now come in and make use of the nitrogen that those plants like alders will produce. This has also been really important, although people in the Middle Ages didn't understand what they were doing, but people discovered nitrogen fixation from leguminous plants by accident. So early farmers realized that if you kept growing crops on the same field year after year after year, eventually your yields went down and down because you were depleting the soil. They didn't realize what was going on, but somebody figured out that if you grew a cover crop or did crop rotation and grew things like alfalfa or clover or peas or beans, these are all leguminous plants. If you just let the field grow with that for a year and then plowed that under, something called green manure. So instead of putting cattle manure on here, which does some good, by putting these plants and letting them grow, they're fixing atmospheric nitrogen, turning it into ammonia. Other bacteria turn it into things like nitrates and nitrites, which allow plants to grow better in the subsequent years. This was actually a very important development in our early history in terms of being able to feed a burgeoning human population in the Middle Ages. But the bacteria, so now let's talk a little bit about the specific genera, plural of genus. Rhizobium, Bradyrhizobium, and Azorhizobium. These are probably the best known, best studied groups of microbes that enter into this mutualistic symbiotic nitrogen fixation interaction. And these are very closely related to the Agrobacterium tumefaciens that we just talked about. They're in the same family. So these are associated with nitrogen fixation, and I mentioned already alder, this plant. You know, those of you especially from the Northeast will recognize alders as growing very much along the sides of streams. So nitrogen in the form of nitrate and things like that is very water soluble. So soils that become saturated very frequently or are associated with running water quite frequently will be rapidly stripped of their nitrate sources. So the best possible plants to grow in those kind of riparian environments, you know, streamside, or in places that are periodically flooded, tend to be things like alders and legumes and things like that, because they can generate their own usable source of nitrogen. So these are what root nodules look like. So this kind of symbiotic mutualistic nitrogen fixation occurs in specialized portions of the plant, usually in association with the roots. So you generally have to pick up a plant out of the soil and shake off the dirt in order to see these things. But these swellings, these hypertrophies or increased cell numbers are the direct result of symbiotic bacteria. So here's comparison and contrast number one between Agrobacterium tumefaciens that causes crown gall tumor and this now mutualistic beneficial interaction with Rhizobium. They both cause this uncontrolled or semi-controlled proliferation of cells. So these are very much like tumors. They don't grow quite to the same size as crown gall tumors, but the same family of bacteria now doing something good for the plants are producing these nodules in association. Now, it's not as if the plants absolutely need these. They can limp by with nitrogen that may be in the soil natively. But if you look at, these are bean plants, green bean plants, side by side, grown in the absence of symbiotic bacteria, so no rhizobium available to form root nodules, as opposed to beans grown from the same seeds at the same time, but now treated with bacteria that are capable of nitrogen fixation. So you can see that growth is much, much better in association with this mutualistic bacteria. So let's talk, so we talked a lot about how Agrobacterium tumefaciens found its way to a plant, started the infection, caused the tumor. Let's do the same thing, but now we're talking about Rhizobium, so this symbiotic mutualistic nitrogen fixation. And what we're really describing here is a dialogue. So a dialogue between the legume plant and the bacterium living in the soil. So they actually, one talks to another and then the other one talks back. So Rhizobacteriaceae are very common, perhaps among the most common bacteria in the soil. And they can live in soil without plants perfectly well. These bacteria, although they're capable of nitrogen fixation, do not need plants. They're just fine without them. But if plants are present, especially if leguminous plants, especially if specific leguminous plants are present, they can enter into this interaction. So what happens is that as a legume, so this is the root of a legume, think of a pea or a bean in your garden. As it begins to grow, this plant, like most plants, releases a lot of complex secondary metabolites into the environment, into the soil. One of the things that they release are a group of molecules known as flavonoids. These flavonoids, I don't know what they really, if there's another purpose for them in terms of the plant biology, but in terms of initiating this dialogue with bacteria, this is the first part of the conversation. So flavonoids are produced by the legumes. They're detected, they diffuse into the soil, and they're detected by rhizobium. So this becomes the chemotactic calling card. So the bacteria will now positively chemotax towards the concentration gradient of flavonoids, knowing at the other end there's a jackpot. There's a plant that it can enter into an interaction with and gain some activity. So these rhizobia that are capable of nitrogen fixation are only capable of nitrogen fixation because they carry a SIM plasma. Again, think about the system of Agrobacterium tumefaciens. The only way that system worked is the presence of the Ti plasmid. It's the same thing with nitrogen fixation. It's entirely driven by a plasmid, a parasitic piece of DNA. This SIM plasmid has all of the genetic machinery necessary to sense the flavonoids, to drive the chemotaxis, and to go through this, I'll use air quotes, infection process that we're about to describe with the plants. It's all about this SIM plasmid. It contains all of the genes necessary for that, including nitrogen fixation. Now, this bacterium, as it lives in the soil, cannot fix nitrogen. Absolutely cannot. Because in the soil, the bacterium is living in an aerobic environment, lots of oxygen. Oxygen is absolutely death for the enzyme nitrogenase that does nitrogen fixation. So as this bacterium exists in the soil with the SIM plasmid, it can't do anything for nitrogen fixation. It can only do it when it gets in association with the roots of the plant. So these free-living bacteria, incapable of nitrogen fixation, will swim their way up this gradient until they find legumes. They'll hit the plant. So they're following towards this. This is the first part of the conversation. The flavonoids will diffuse across the cell envelope. So you can tell by the ring structures of these that they're pretty permeable through things like lipid bilayers. So with no active transport necessary at all, these flavonoids gain access across the envelope of the rhizobium and now have the ability to do something very similar to cyclic AMP binding to the CAP protein that we talked about in the last section for the last exam. When these flavonoids bind to a protein called NOD-D, NOD-D is a transcription factor. It's encoded on the SIM plasmid. So again, this is all about the SIM plasmid. This NOD-D protein, without being bound to the flavonoids, is completely inactive. It does nothing. But when it's bound to the right flavonoid, when the right molecule, the exact right molecule, fits into this NOD, it now is allosterically modified, and it will induce a gene expression change in the bacterium. So the conversation has been heard, it's been acknowledged, and now the bacterium is preparing to respond in this dialogue. So the flavonoids are inducing the expression of several genes, one of which is a protein on the outer membrane of this gram-negative bacterium called Ric adhesin. You can tell from this suffix adhesin, this is a protein that's going to be involved in allowing the bacterium to bind to the legume once it finds it. So as the legume is swimming up this gradient looking for the source of the flavonoids, it's beginning to change its gene expression. It's beginning to decorate its coat, if you will, by putting on outer membrane proteins that it didn't have before. It's dressing up for this meeting with the plant. It's putting out Ric adhesin proteins onto its outer membrane so that when it encounters a root hair like this, it has the ability to bind to it. So this infection process begins because now Once the bacterium encounters this root hair and binds to it via these Rick adhesins, it answers, it continues the dialogue. So now the conversation goes from the bacterium to the plant cell. The bacteria are going to produce soluble factors that are going to be secreted called NOD factors. NOD here is, so I should have mentioned this before, NOD you can think of, this is short for nodulation. So these are factors that are going to initiate those nodules, the root nodules where nitrogen fixation has occurred. So we call them NOD factors. They're produced by the bacteria, secreted, and they're going to be picked up by the plant cell, and the plant now knows, okay, my conversation has been heard, the bacteria have gotten here, and now they're knocking at the door. They're telling me they're here, so it's time to change the plant gene expression. So the flavonoids of the legume enter so the NOD factors from the bacterium enter the legume root, and they change the gene expression of the plant. So it's a true dialogue. Now, these NOD factors that are produced by the bacteria have a chemistry that's not unfamiliar to you. So you remember from our discussions on peptidoglycan the importance of N-acetylglucosamine. Well, that's what makes up these NOD factors. So as a general plan, so every one of these bacterial species that produces these things produces very specific NOD factors. But in terms of the general characteristics of all NOD factors, they're generally between 3 and 5 polymerized N-acetylglucosamine molecules that have all kinds of different substituents on them. So one specific species might have only one methyl group here, or it has a slightly different substituent here. Because what happens is these actually fit like lock and key. Not every bacterial rhizobial species can colonize, or in fact, every single legume. These things are very highly evolved. There is a specific bacterial species that will colonize beans. There's another one that will colonize peas. Any of you or your families who are into gardening may realize if you look through certain catalogs, you can buy bacteria to introduce into your soil to help your beans or peas fix nitrogen better. But there are different species. So you can get specific species that will help your beans grow. But those bacteria don't do a very good job, if at all, with peas. Part of the reason for that species specificity is these NOD factors, and the flavonoids for that matter. So only bacteria will respond to only certain key flavonoids, and then they'll produce one specific NOD factor. One of the things that's common in all of these NOD factors is the presence of one substituent, which is always a fatty acid. This fatty acid is what allows this NOD factor, regardless of what other substituents it has, this fatty acid gives enough hydrophobicity so that the NOD factor can get access across the plant cell membrane. So these NOD factors diffuse into the cell and affect gene expression. So there's another means, so these systems are very highly evolved, and they can help stop competing bacteria from infecting them or even proliferating. Because sometimes the flavonoids from certain legumes will bind to the NOD-D protein of the wrong species. So of a species of bacterium that might compete with the bacterium that they want. Flavonoids from peas, let's say, have the ability to bind to the NOD-D of the bacterium that would colonize beans. And when that happens, they'll actually inhibit NOD factor production in those bacteria. So those bacteria can't carry on a conversation. They can't answer back. So it's a way to make sure that only the correct bacterial species finds access to these plants. So one of the things that happens in terms of gene expression in the plants in response to the proper NOD factors is that they'll form what's known as a shepherd's hook. So when the bacterium is attached via its Rick adhesins and the proper NOD factors are now taken up by this plant, the gene expression changes so that now that root hair will form a shepherd's hook. It will basically, engulf. It'll give a big hug, if you want to think of it in warm, friendly ways, to this bacterium. So this is all about gene expression changes within the plant. The gene expression changes will continue. So as long as the bacterium is producing the right NOD factors, then the dialogue goes forward. The intimacy of this relationship proceeds to the next level. So what will happen next is that within that shepherd's hook, there will be formed a cellulose tube, which will allow these motile bacteria to now infect, if you will. I think infect has a bad connotation, right? In fact, we all try to avoid infections as best we can. But this is actually a beneficial infection. You can think of it as maybe colonization is a better word. Colonization sounds more friendly than infection. But in either case, this is actually called an infection thread. So the bacteria will swim down through this cellulosic infection thread and gain access to the cytoplasm of the plant cell, but not completely. So this bacteria, or this plant is being very cagey, it's being very careful. It doesn't just let these bacteria have free access to the cytoplasm. At the end of this infection thread, it pinches off the bacteria, it lets the bacteria into the cytoplasm, but it puts one of its own membranes around it. So it's maintaining some degree of separation between this now symbiotic, this nascent symbiotic bacteria. It trusts it, but not completely. So this is called a parabacteroid membrane, this plant-based membrane that surrounds the bacterium into an intracellular vacuole called a symbiosome. Very cool, very appropriately named little bag of bacteria. These bacteria will now respond. So now they have the ability to sense that their environment has changed, that they're within a parabacteroid membrane, and they will undergo differentiation. Not just in terms of gene expression, but they actually undergo a morphologic differentiation. So many of these rhizobia turn into these Y-shaped bacteria that have concomitantly lost their ability to reproduce. These bacteria are now irreversibly committed to symbiosis. They are completely domesticated. You can release them from this environment now and they just will not grow. They've undergone a terminal differentiation. But it's only now in this particular situation where you have the bacterium intracellularly and within a parabacteroid membrane that nitrogen fixation has any chance to proceed. Up until this point, there is still no nitrogen fixation going on within the plant. But now the situation is set up where that actually can occur. So this is the process now of nitrogen fixation. So once it's inside the symbiosome, although the nitrogenase enzyme can be expressed, so remember that the symplasmid, which is not shown in this cartoon, encodes the enzyme nitrogenase, which is capable of taking dinitrogen, N2, and reducing it to NH3, the biologically usable form. That's encoded by the symplasmid. It can be expressed in here, but it won't be functional. Nitrogenase, the enzyme, is exquisitely sensitive to oxygen. Even small amounts of free oxygen will kill that enzyme. So, but here's the conundrum, because the bacterium itself is obligately aerobic. It cannot exist fermentatively. It cannot use any other terminal electron acceptor other than oxygen. And you obviously know that the plant needs oxygen. They're obviously aerobic. So these two things don't seem to fit together. You have an enzyme which can't function to be around oxygen, involved in two different bacteria that have an absolute requirement for it. So what happens is that the plant-legume bacterial interaction sets up an environment within the symbiosome which is microaerophilic. So in other words, there's very little, tremendously little free oxygen. They do that by producing an oxygen scavenging protein called leg hemoglobin. Leg obviously coming from the fact that this is a leguminous plant protein. And hemoglobin obviously tells you that this is capable of binding oxygen. Leg hemoglobin, what this does, shown here at the bottom of this cartoon, it binds oxygen very, very tightly. And it has the ability to deliver that oxygen in a bound form, not in a free form, but in a bound form, to the end of the electron transport chain of the bacterium. So what happens is that as the electrons pass through the electron transport chain, They can reduce oxygen to water because oxygen is being supplied in the bound form, not in the free form as is occurring in you and I. This allows this symbiosome to have vanishingly small oxygen levels, free oxygen levels. And it's because of those vanishingly small oxygen levels that nitrogenase can actually be expressed and reduce atmospheric nitrogen to the usable form of ammonia. Now, the really cool thing about this symbiosis in terms of this leg hemoglobin is even this molecule is a symbiotic product. The heme portion of leg hemoglobin, that portion that has the iron and binds oxygen, is actually synthesized by the bacterium. Yet the protein is synthesized by the plant. So it's a joint venture to synthesize this molecule. It's then put together for the benefit of both the plant and the bacterium. And if you look at root nodules, oftentimes you can see the red color. So this hemoglobin is iron-based, just like ours. So for the same reason that our blood is red, you can see red color in the root nodules of certain legumes if you look at them. So now, how is it that this symbiosis works? It's really not based on the fact that nitrogenase is occurring. What's happening is that the plant, being a good host that it is, having invited these bacteria in, need to feed it. They need to provide it dinner. So what they do is they take some of their photosynthate in the form of sugars, and especially in the form of organic acids, and they supply that to this now domesticated bacterium so that things like succinate, malate, and fumarate can feed directly into the citric acid cycle. Glycolysis really stops at this point, and the bacterium is largely producing all of its energy via the citric acid cycle. So it's producing massive amounts of ATP, because that's the other really kind of bad thing about nitrogenase, is it requires a huge amount of ATP. The process of nitrogen fixation energetically is tremendously expensive. But all of the ATP that's being generated, not all of it, but some of the ATP that's being generated here is going into nitrogenase, which is turning atmospheric dinitrogen into ammonia, which the bacteria will now use to produce amino acids. It will also take some of those amino acids and transport them into the plant in the form especially of the amino acids glutamine and asparagine. This is largely where most of the nitrogen comes from, the usable nitrogen of plants, because plants will now take glutamine and asparagine and actually turn them into all of the other amino acids. There is some degree of ammonia being directly transferred to the plant, but I think most of the usable forms of nitrogen that are coming from the bacterium to the plant are in the forms of these amino acids. The nitrogenase itself, as I mentioned, is very energetically expensive. It takes 16 ATPs and a bunch of reducing power, so in the form of NADPH, these electrons are coming in. So nitrogenase has the capability in multi-step process. So this is a little bit unusual biochemically because it takes several steps for nitrogenase to produce as the end product NH3, ammonia. But there is no release of an intermediate. So there is, the first reduction occurs to this atmospheric dinitrogen, but this N2H2 molecule remains bound. It will be reduced yet again, so more hydrogens are added to it, yet it's not released. It's not until this third reduction occurs where now the nitrogen is fully reduced to ammonia that the ammonia is actually released from that cell. So this is not only taking a lot of ATP, but it's taking a lot of reducing power to produce this. So this is part of the reason why this only occurs in the presence of the plants, because now the bacterium is in a situation where it has a constantly solid source, a reliable source of energy in the forms of these organic acids that the plant is providing to the bacterium. Because of that, lots of food, it has the ability to do lots of work. And to pay for that, it's providing ammonia and amino acids to the plant. The nitrogenase enzyme itself is really interesting too. because it actually has an iron molybdenum center. So as you look at biochemistry, a lot of times when you see things that deal with nitrogen, nitrogen reduction, nitrogen oxidation, you'll see that molybdenum is obviously very important. But we don't get to talk about molybdenum very often in biology. And I even just like saying the word molybdenum because I have to practice that on my drive in because I could really stumble over that word. But it's a really interesting enzyme, and it takes a lot of electrons and a lot of reducing power in order to make that. Now, question, yes, Jess. Good question. So where does the NADPH that's generating these electrons for these reductions come from? I am assuming that it's coming from the TCA cycle of the bacterium, but I don't know that for sure. But that would be the most likely source for the reducing power. Now, that is a tour de force through nitrogen fixation. I realize that. But this is only a survey course. People could teach an entire class in nitrogen fixation and the bacteria that mediate it. But what you hopefully see is that if this process were just allowed to go forward, eventually all of the atmospheric dinitrogen, which is 80% of our atmosphere, would get sucked out of the environment and would now be in the forms of organics, in the forms of ammonia or nitrate. So now I want to kind of transition from this symbiotic mutualistic comparison of rhizobia and agrobacterium and talk a little bit about the importance of bacteria in terms of global mineral cycling. So, and since we're talking about nitrogen fixation, this is a good example or a good jumping off point to talk about the nitrogen cycle, and because it's bacteria that drive all of the major points in the global nitrogen cycle that keeps things going through. So the atmospheric dinitrogen that we just were talking about can be done by, oftentimes by soil nitrogen fixers, as we just talked about, to generate ammonia. And then in soil environments at that pH, it becomes typically an ion, the ammonium ion. But now, rather than just have all of that nitrogen, ammonia buildup, there are bacteria called nitrifiers, which can now take ammonia and use it as an energy source. So if you think about this, this is a highly reduced form, right? When you see an atom like this with 4 hydrogens attached to it, that's highly reduced. So that's very much like a carbohydrate to you and I. So while you and I find glucose very delicious and a source of electrons or energy, there are chemolithotrophic bacteria in the environment that can oxidize, and we're going to talk about them ad nauseum, that can take ammonia and oxidize that to things like nitrite and nitrate, which, as it turns out, are much better biological sources of nitrogen for plants. Typically when you fertilize your gardens at home, if you're fertilizing them with nitrogen, you're typically going to add nitrates. More commercial agriculturalists, farmers, tend to put ammonia in because ammonia will stay around longer. But this form is actually more utilizable by plants. But again, all of the nitrogen would get sucked up from the atmosphere and turned into nitrate. So there are bacteria that can now use nitrates as terminal electron acceptors. We've already talked about this. E. coli does this. Your favorite friend E. coli is reducing nitrates to nitrites. There are other bacteria that will reduce nitrites further to nitric acid, to nitrous oxide, and eventually all the way back to atmospheric dinitrogen. So there's this cycle of nitrogen going through the environment, all driven by bacteria. Now, the process of nitrification, so I want to talk about first, is where ammonia, we just talked about the generation of ammonia in nitrogen fixation, is now turned into nitrate. That's the top half of this circle. The process where under aerobic conditions where two certain groups of bacteria will collaboratively take ammonia all the way to nitrate. This entire process, shown on the top, is by definition nitrification. NH3 to NO3. But that process, by the way, has to occur aerobically. All of the bacteria that are doing this under aerobic or oxygen conditions. The first half of this condition is done by bacteria which very conveniently taxonomists always put the prefix in front of called, do I have it written here or just here? Here it is, nitroso. When you see the prefix nitroso in front of a genus like this, Nitrosomonas, that means that that bacterium can oxidize ammonia into nitrite. That process, so that's shown here from about 3 o'clock on this to 12 o'clock, from ammonia to nitrite. That process, that sub-process of nitrification is called nitrosification. By definition, nitrosification is taking ammonia to nitrite. Bacteria like Nitrosomonas are very good at doing that. They're using this as an electron source. Ammonia is their food. It is their energy. It's not their carbon source, obviously, but it is their source of electrons and energy. And they oxidize that to NO2. This is an important point, there is no bacterium known that can take ammonia and oxidize it to nitrite and then further to nitrate. Bacteria can either oxidize ammonia to nitrite and no further, or they can take nitrite and oxidize it to nitrate. But those bacteria can't do the first half of that. So this whole semicircle on the top here, this nitrification, is another example of my favorite word in all of biology, syntrophy. You have one group of bacteria, such as the Nitrosomonas, which are capable of nitrosification, taking ammonia to nitrite. But then nitrite is actually toxic to the bacterium. It's an end product of that metabolism. So if it builds to too high a level, that bacterium will suffer. It won't be able to oxidize ammonia any longer. So it has to live in association with bacteria that will take the nitrite and reduce it to nitrate, finishing the process of nitrification. That's actually done by groups of bacteria that always have the group or the prefix in front of them, nitro. Nitrobacter is a genus of bacteria that will take nitrite, NO2, and oxidize it to NO3. These two groups of bacteria cannot live without one another. So that completes this top half of the nitrogen cycle on the top, nitrification. And when we come back on Monday, we'll continue this wonderful story of the nitrogen cycle. Have a great weekend, and I'll see you guys in the lab, or not.